Okay, I guess you can start, right? We have mostly all the students. Yeah, I'm audible.
Yes, ma'am. Ma'am, can we start now?
Yeah, you can start.
Okay, ma'am. So good afternoon, everyone. Today our group is presenting on the topic Begumoviruses infecting vegetables in India. So, speaking of Begumovirus, it is a genus of single-stranded DNA virus in the family Geminiviridae. It is the largest genus consisting of 445 species as per the last data released by ICTV in 2020. The name Begomovirus is obtained from bean golden mosaic virus. They have a unique morphology as they are typically twinned, or we can say paired with two incomplete icosahedral symmetry, as you can see in the figure on right side. Each twinned particle contains only a single circular, single-stranded DNA. There are 22 capsomeres per nucleocapsid, and each nucleocapsid is 38 nanometers long and 32 nanometers in diameter. The genome size can range from 2.6 to 2.8 kilobase pairs approximately, and the genome can be monopartite as well as bipartite. You can see the diagrammatic representation below. So monopartite means it has a single genomic component which is identified as DNA-A. Bipartite genome means that the genome is segmented into two segments, referred as DNA-A and DNA-B, that are packaged into separate particles. The DNA-A component encodes all virus factors required for control of gene expression, genome replication, and insect transmission between hosts. The DNA-B encodes a protein involved in intra and intercellular movement in host plant tissues. Both the segments are generally required for successful symptomatic infection in the host cell, but DNA-B is dependent for its replication upon DNA-A, which can, in some begumoviruses, apparently cause normal infections on its own. They are also associated with alpha or beta satellite DNA, which has role in symptom development and virulence. Begumoviruses principally affect the dicotyledonous plant species. At the right corner of the slide, you can see the electron microscope view of Begumovirus virions, which beautifully shows the twin icosahedral symmetry. Then moving to the virus classification, Begomoviruses belongs to group 2 as per Baltimore classification, and according to ICTV, the International Committee on Taxonomy of Viruses, the classification of Begomoviruses follows as realm monodNA viria, kingdom Shotoku phylum DNA viricota, class Repensiviricities, order Geplavirals, family Gemini Viridae, and genus Begomovirus. So if we talk about the host, then Begomoviruses have been known to be associated with and cause many diseases in cucurbitaceous, solanaceous, malvaceous vegetables, and legume crops. These viruses have emerged as a major threat to vegetable and legume production in India. Some examples of cucurbitaceous vegetables are bitter gourd, pumpkin, ridge gourd, which we call torei in Hindi, ash gourd, also called as wax gourd, ivy gourd, etc. All are associated with a yellow mosaic disease virus. Examples of solanaceous vegetables are chili, which is associated with chili leaf curl virus, potato, which is affected by potato apical leaf curl virus, tobacco, in which tobacco leaf curl virus and tobacco ring spot virus cause disease. Brinjal, also known as eggplant, and tomato, they are associated with tomato leaf curl viruses. Examples of malvaceous crops include okra, also called as bhindi, in which the disease is caused by Bhindi yellow vein mosaic virus and Okra initiation leaf curl virus, and cotton associated by cotton leaf curl virus. Legumes are the richest source of vegetable protein, and different types of vegetable legumes like cowpea, dolichos bean, common bean, and soybean, etc., are occupying a major area in India and constitute the major dietary constituent. These crops suffer losses due to Begumoviruses like mung bean yellow mosaic virus, dolichos yellow mosaic virus, tobacco curly shoot virus, etc. Other crops affected by Begumoviruses are radish, which is affected by radish leaf curl virus and Croton yellow vein mosaic virus, cabbage, which is associated with cabbage leaf curl virus, and cassava, in which Begumoviruses like Indian cassava mosaic virus, Sri Lankan cassava mosaic virus cause disease. All the diseases and symptoms of Begumoviruses will be later discussed in the slides. The following map represents the distribution of Begomoviruses around the world. So we can see that the tropical and subtropical regions are majorly affected by these viruses. Now let's see the distribution and diversity of tomato Begomoviruses in India. Leaf curl Begomoviruses cause serious yield losses to Indian tomato crops. Different states all over India affected by various species are represented on the map. Monopartite Begomoviruses species include tomato leaf curl Kerala virus, tomato leaf curl Ranchi virus, tomato leaf curl Patna virus, tomato leaf curl Rajasthan virus, tomato leaf curl Pune virus, tomato leaf curl Lucknow virus, and so on. Bipartite Begomoviruses include tomato leaf curl New Delhi virus and tomato leaf curl Palampur virus. These tomato leaf curl viruses have a wide host range and infect tomato, eggplant, cucurbits, cotton, chili, cowpea, okra, and tobacco crops, as well as some common weeds. In this slide, the first map represents the distribution of Begomoviruses infecting okra, bhindi, exhibiting yellow vein and leaf curl disease in India. We can interpret that bhindi yellow vein beta satellite in blue dot occurs majorly. In contrast, okra leaf curl beta satellite in red dot appears to be limited to northern India, whereas Croton yellow vein mosaic beta satellite in yellow dot appears to have a host range running from northern states to the northeastern coastal states of West Bengal and Orissa. The map in center represents the distribution and diversity of Begumoviruses causing chili leaf curl disease across different states of India. Chili leaf curl disease causes severe crop loss in the tropical and subtropical regions of the Indian subcontinent and has emerged as a major factor limiting chili cultivation. Also, Indian cassava mosaic virus causing disease in cassava, which is an important food crop, has been reported in India from following states: Andhra Pradesh, Karnataka, Kerala, Madhya Pradesh, Maharashtra, and Tamil Nadu. Let's now move ahead and find out how begomoviruses are transmitted and cause infection in the host plant. So, begomoviruses are obligately vectored by an insect vector called whitefly, Bemisia tabasi, conventionally known as the whiteflies or silverleaf whiteflies. This species thrives in temperate habitat worldwide and cannot survive in cold environment. This vector allows rapid and efficient propagation of the virus because it is an indiscriminate feeder. Female bee tabasi lays 100 to 150 eggs on either side of the leaf. The color of eggs is yellow or orange and are minute cigar in shape, which is laid in groups. Hatching occurs in 2 weeks with nymphal instars. Those are pale yellow in color. From pupae, adult formation takes place after 1 week, which is white in color. On an average, These flies give rise to 11 generations every year. Also, the vector whitefly transmits the virus in a persistent, circulative, non-propagative manner. We all are familiar with these terms, but if in case you guys don't remember, then persistent means that the viruses are retained in insect guts and can spread to salivary glands. Circulative, non-propagative manner is that the viruses do not replicate in vector tissues but move from insect gut to hemolymph and further to the salivary gland, from where they are released into the plant tissue during feeding. Now Yukta will discuss about the life cycle of Begumoviruses from the next slide.
Thank you, Ayushi. Now, moving to the next topic, life cycle of Begomoviruses. So just to give an idea how the infection start, I've attached this picture showing vector-mediated transfer of the virus particle and thereafter the seed-mediated transfer by the infected plant. So whitefly carrying the geminate particle or the virus particle upon uncoating releases the viral single-stranded DNA to the infected plant cell cytoplasm, and the viral DNA is imported to the nucleus for the amplification. So the single-stranded DNA gets converted to double-stranded using the host cell machinery, and the viral DNA is in association with the nuclear proteins, which I'll be mentioning, can transcribe viral mRNAs at the nucleus, and later ones are exported to the cytoplasm for translation to the different viral proteins. The viral proteins that are required in the nucleus to carry out replication, transcription of or movement of viral DNA enter to the nucleus through the nuclear pores, and with the help of movement proteins, the DNA can enter into nearby cells through plasmodesmatal channels, and thus this is how the virus spread occurs. It will be more clear in the next slide. Next slide, please. Yeah, thank you. So when the silver leaf white fly interacts with the, I've taken here the example of infection in cotton plant. So when the whitefly interacts with the cotton plant leaf, the transfer of the geminate particles take place. The single-stranded DNA of the cotton leaf curl virus is transferred into the cytoplasm. From there, the single-stranded DNA enters the nucleoplasm via single-stranded DNA NSP complex. NSP here is a non-structural protein that helps in the entry of the viral DNA. So this complex is formed when the cotton leaf curl virus single-stranded DNA interacts with the nuclear shuttle protein, and the single-stranded DNA undergoes replication by involvement of the viral replication protein, or we can say rep protein, and the virus replication accessory protein, which we can say REN or REN protein. So this gives rise to the double-stranded DNA replicative form. In short, I'll be calling it RF, the replicating form. So the RF acts as a template for the replication and transcription, which can be methylated or non-methylated. So this replication undergoes by the rolling circle mechanism, the transcription activator protein, which we can also call as TRAP, transcription activator protein, and the viral replication protein, RAP, helps the double-stranded DNA undergo transcription for the synthesis of viral mRNA. Then this mRNA then codes for several viral proteins like Rep, REN, ACE123, etc. So also the double-stranded DNA, the replicative form, when turned into the single-stranded DNA, it interacts with the NSP to form again the single-stranded DNA NSP complex, which shuttles the viral single-stranded DNA in between the nucleus and the cytoplasm. So in the cytoplasm, this complex interacts with the movement proteins, which transports this complex towards the periphery of the cell and helps it to move across the cell wall, which causes infection in the surrounding cells as well. So when this single-stranded DNA of the cotton leaf curl virus interacts with the coat protein, it forms the capsid of the virus particle. Then this protein checks the virus transport through the intestinal wall of the white fly, as Ayushi mentioned, into the hemocoel, where the coat protein binds with the molecular chaperone produced by endosymbiont bacteria to form a complex, which is known as CP-GROL complex. So this complex provide protection to the viral DNA from degradation inside the vector. Moving to the next slide. So why Begomoviruses infect so many plant host species? So the Begomoviruses may be transmitted by their vector, in, the white fly, in genetically diverse hosts. So due to various environmental climates and geographical niche, the genotype of the plant and the vector may differ. So virus replication is one reason, and the selection within the host is a continuous process. So during this process, the interaction of begomoviral proteins with the proviral and antiviral proteins regulate the balance between the variation and the selection, leading to the selection of the fittest or the most adapted strain of the virus particle. So vectors contribute to the selection made by transmitting the virus in the new plant species or different genotypes or cultivars of the same species. Some begomoviruses retain a satellite DNA called beta satellite, and this interaction is known as begomovirus beta satellite complexes. So these beta satellites depend on the helper virus for the replication and spread within and between the hosts. So the selection, also the selection pressure enforced on a virus genome by a given environment can also alter the viral population. And the last reason is the mutation that offers a beneficial advantage to are probably to be fixed in the genome, and some mutation generated in alternate host might also break the resistance and even expand the host range. Moving to the next slide. Symptoms and the disease caused by begomoviruses in India. I'll be mentioning that. So the table number 1 includes the tomato yellow leaf curl virus, which causes the disease leaf curl. And the symptoms can be seen as curling and puckering of leaves, chlorosis, and stunting of the plant. Next is bitter gourd yellow mosaic virus, which causes the disease mosaic, and the symptoms are yellow mosaic and chlorosis. Next is mung bean yellow Indian mosaic virus. It causes the disease yellow mosaic in the plant, and the symptoms along with the yellow mosaic are curled leaves and stunting. Another one is reddish leaf curl virus, which causes leaf curl, and along with that is the symptoms are stunted and distorted growth. Next is Croton yellow vein mosaic virus. It affects radish. It also affects radish, like radish leaf curl, but it causes mosaic in the plant. Or it also the symptoms also include leaf curling, yellow mosaic, annation on the lower side of the leaves if the cases are severe. Next is Dolichos yellow mosaic virus. It can occur on cowpeas, Dolichos beans. It causes the disease golden mosaic disease, and the symptoms are golden yellow mosaic patches on the leaves of the infected plant and stunted growth. Moving to the next slide. So next species of Begomovirus is tobacco curly shoot virus. It causes the curly shoot disease. The symptoms are stem twisting, stunting, curly shoot, and thickening of the lower leaf surface veins and galling with the dark green color on the leaves. So next is papaya leaf curl virus. It causes the leaf curl disease. The symptoms are curling, crinkling, and distortion of leaves, reduction of leaf lamina, rolling of the leaf margin inward and downward, and also the thickening of veins. Leaves become brittle and distorted, and the plants are also stunted. So affected plant does not produce flowers and fruits. The next is horse gram yellow mosaic virus, which causes the disease mosaics. The spots gradually increase in the size, and ultimately the leaves turn completely yellow, and the infected leaves also show necrotic symptoms, and the diseased plants are here also stunted and they mature late and produce very few flowers, and pods of these infected plants are also reduced in size, and they also turn yellow in color. Next is Sri Lankan Cassava mosaic virus, which causes the disease mosaic. The symptoms are mosaic pattern, stunting, leaf curl, and chlorosis. The last is squash leaf curl China virus, which causes mosaic of cucurbits like pumpkin, bitter gourd, squash, ash gourd. The symptoms are mosaic mottling on leaves and reduced leaf size, leaf crinkling, distortion, and yellow mosaic patches on the leaves. Now, Tanvi will further discuss about the common symptoms caused by Begomoviruses in detail.
So I will So I will start with symptoms. So let's just go through some terms used for symptoms, which will help us to differentiate between the two symptoms. So here I have mentioned some name terms for the symptoms. So first is the stunting and dwarfing, that is overall impact on plant size. Then there is color deviation in leaves. So first is mosaic. It shows irregular patches of colors, that is dark green or light green, or it can be yellow. And mottle, it is evenly distributed patches of light and dark green on the leaves. And variegation is patches of chlorotic green or other colors. And vein flecking is a water soaked effect on the back of leaf corresponding to vein cleared areas. Vein bending is dark green bands are seen along with the veins. And vein yellowing is vein and veinlets turn yellow. And in yellow mosaic symptom, we can see that the entire leaf turn golden yellow. Next slide. In this, there is epinesty symptom, which shows that leaves curl or turn downwards. Then hyponesty is opposite of that. So in this, leaf curl or turn upward. In inhalation, outgrowth on leaves from veins of lower surface can be seen on leaves. Blisters are like epidermis of leaf forms raised bubbled like growth. And in cupping, cup-like depressions on leaf can be seen. And in twisting, leaves and petioles get twisted. And then malformation, imbalanced development, that is hormonal disturbance. So in this, leaf rolling can be the symptom. So in leaf rolling, leaf curling and leaf distortion can be seen. In leaf narrowing, the fern leaf and shoestring effect can be seen impeded growth of laminar tissue. Rugosity is the impeded growth of venal tissue, and rosetting is the impeded internodal expansion at stem tips. So these were some terms which are used for symptoms, and we can differentiate between the two symptoms. So now I will start with symptoms of Begomovirus. The first is the Bhindi yellow vein mosaic virus. It's in this yellowing of the entire network of veins in the leaf blade is the characteristic symptom. And in severe infections, what happens is that the younger leaves turn yellow and it becomes reduced in size, and the plant is highly stunted. The veins of the leaves will be cleared by the virus, and intervenal area becomes completely yellow or white, as I have shown in the picture. So, in a field, most of the plants may be diseased, and the infection may start at any stage of plant growth. So, infection restricts flowering and fruits. If formed, if fruits are formed, then maybe it can be smaller or harder. And the if affected plants produce fruits with yellow or white color. So and so they won't be fit for marketing. That next slide. Then is the Okra initiation leaf curl disease. In this disease was found in Karnataka. It was observed there, and as curling and upward cupping of leaves can be seen, and reduced leaf area along with initiation and petiole bending, and the resultant plant showed stunting, reduced yield, deformed fruits, unsuitable for marketing, and it was in severe condition. A yield loss of 80 to 90% was seen there. So this disease was named as Okra initiation leaf curl disease. Next slide. This is the chili leaf curl virus. So, in this, chili is an indispensable ingredient of Indian cuisines that we all know. And leaf curl of chili is characterized by shortening of leaves and stem, or vein thickening, or vein clearing, and curling of leaves. So the infected plants reduce yield and produces few flowers and fruits only. So the four strains was reported from India, and the virus can be transmitted to four different plant species of Solanaceae family. For example, Capsicum annum, Solanum lycopersicum, and Nicotiana benthamiana, and Nicotiana tabacum. These were the four different species from which virus can transmit. Next slide. This is the Indian Cassava mosaic virus. So in this, we see that mosaic symptom reduction in leaf size was observed, mishappened, twisted leaves were seen, and the affected plants stunted, tuber splitting, and yield reduction was seen. The virus spread by the infected sets and white fly, that was Bemisia tabaci. Next slide. This is the tomato leaf curl virus. So, in this, symptoms were like the new growth of plants with tomato yellow curl has reduced internodes, giving the plant a stunted appearance. The new leaves are also greatly reduced in size and wrinkled. are yellowed between the veins and have margins that curl upward, giving them a cup-like appearance. So flowers may appear, but usually will drop before fruit is set. Next slide. This is the mung bean yellow mosaic India virus. So in this virus, the yellow characteristic symptom is like bright yellow mottling of leaves, and the yellow area are scattered or occur in indefinite bands along the major veins. So rusty necrotic spots appear in the yellow areas as the leaves mature, and pulses are the major sources of protein in the vegetarian diet in our country, that we all know. And besides being a rich sources of protein, they maintain soil fertility through biological nitrogen fixation, and it plays a vital role in furthering sustainable agriculture. So among the pulses, mung bean is one of the most important grain legumes that is grown particularly in Asia. And due to its protein rich edible seeds, its ability to fix nitrogen or drought tolerance and early maturity, it is widely planted in various cropping systems. So I have shown in this figure also that first is the resistant leaves, then there is some yellowing, light yellowing, then moderately resistant, then moderately susceptible, then it is susceptible, and then in highly susceptible it becomes yellow. Next slide. This is the cotton leaf curl virus. So the disease generally requires host plants to be infected with both a cotton leaf curl virus and an associated beta satellite component in order to produce symptoms. So plants that are infected with the cotton leaf curl virus but not the beta satellite may appear symptomless. So, symptoms of cotton leaf curl disease on infected cotton plants usually appear within 2 to 3 weeks. And early symptoms include deep downward cupping of the youngest leaves by swelling or darkening of leaf veins. And in later stages of infection, it involves the upward or downward curling of leaf margins. Uh, that is shown in the first figure, that is accompanied by leaf yellowing or mosaic. In the second figure, cup-shaped leaf-like structures, innations can be seen that may form along leaf veins, and it can be typically on the underside of the leaves. And early or severe infection of cotton leaf curl disease may lead to stunted plant growth, and infection while the plant is still young can significantly impact flowering, ball formation, maturation, yield, and lint quality. Next slide. This is the potato apical leaf curl virus. So in this, apical leaf curl disease has emerged as a new disease in potato during the last decade in India due to a change in planting date and an increased whitefly population. So its incidence is on the rise, threatening the cultivation of potato across the country. So a PCR assay was developed for the detection of tomato leaf curl nudity virus, which is the casual agent of apical leaf curl disease in potato. So the affected plants show curling, crinkling of apical leaves with a conspicuous mosaic symptom. And when the infected tubers are used for planting, the entire plant shows symptoms with severe stunting due to high virus titer. Now, Vivek will tell us about the management and losses of Begomoviruses.
Am I audible?
Yes, Vivek, you're audible.
Uh, yeah, my part was the losses on Begumoviruses. Could we move to the next slide? Just kind of just going over some basic facts which have already been explained by my partners. Gemini Viridae is the family which consists of a maximum number of viruses, and a Begomovirus is the largest plant virus genus in the Gemini Viridae family, infecting a wide range of monocots and dicots and causing severe damage. Now, when it comes to losses due to the Begomo viruses, we can see that Gemini viruses, they were actually found to be responsible for 18 to 100% of the yield loss, depending on the host environment and the type of the virus. And in fact, during the 1950s, Begomo viruses, they were blamed for around 20 to 100% of the significant losses in vegetables. Um, Also, Begomoviruses, they have caused more than $300 million in losses in the bean industry in India. So that includes crops such as the black gram, mung bean, soybean, etc. Next slide. Now, Begomoviruses, they have been known to infect a wide range of crops, including agriculture crops, vegetables, medicinal crops, ornamental crops, weeds, etc., with a huge economic loss affecting common people. And the rapid evolutionary potential of these single-strand DNA viruses combined with the polyphagous feeding behavior of whiteflies, it can lead to the emergence of devastating viral strains. So because of this, Begomoviruses, they drew more attention globally by researchers to establish any host pathogen interaction or vector host relationships, the characterization of viruses, the virus reservoirs, phylogenetic relationships, roles of the viral genes, and management strategies. Now, we can look at several examples of certain Begomoviruses, and while I've just provided two, um, Solanum nigrum and Tagetes patula, these are both two different types of Begomoviruses. They both play a major role as alternate or reservoir hosts for the transmission of the disease to other economically important crops. So we can see that these plants not only lead to the infection of one plant, but a multiple number of plants. So that's why it's dangerous. And then another example, another plant, another Begomovirus is the Papaver somniferum, which basically it does 100% infection on opium poppy plants, and it imposes threat to other medicinal important crops. So while these are just two three examples, there are lots of plants, lots of Begomoviruses which have dangerous effects on plants, several plants. Next slide. Now, when we're talking about Begomoviruses, it's important to look at any new disease breakouts in India. So actually, there was a review article that was done. Basically, researchers they took several different Begomoviruses and conducted their sequence analysis, finding genetic similarities to other Begomoviruses, which proves the invasion of foreign species into India. So, as an example, we can see that a variant of tomato leaf curl Bangladesh virus was found infecting Giardia. Now, analysis using the BLAST program revealed nucleotide sequence identities of 94% with tomato leaf curl Bangladesh virus and 92% with chili leaf curl virus TU. Moreover, a Begomo virus infecting Menthas picata was also reported in Lucknow and in the Tarai region of Uttaranchal province. Its sequence showed 93% identity with the Begomo virus tomato leaf curl Pakistan virus. Now, basically, seeing that certain viruses in India, which are being found, have sequence identities with viruses in other countries that we have seen, that goes on to show that yes, there has definitely been an invasion of foreign species in India. Um, Another example of this was the chili leaf curl Velanan virus found infecting chilies in the Velanad region of Kerala, India. And this virus, it had the highest levels of nucleotide sequence identity with a Pakistan strain of the pepper leaf curl Bangladesh virus. And that's not it. I mean, there's several examples that we could talk about. There is an example of sun hemp, which is extensively cultivated in India, which was being found infected by an Indian tomato leaf curl virus, and the sequencing was done on that plant, and the identity of it was seen that it was the same of 92% with another virus found in Pakistan. So definitely we can see that a lot of new disease breakouts have been occurring majorly due to the invasion of foreign species in India. Next slide. Now, there was another case study done recently in 2021 on the losses due to Begomoviruses. So this research in 2021 was based in Chhattisgarh and was done upon three diverse archaeological zones. Now, intense cultivation, climate change, and host pathogen dynamics interactions had led to the emergence of viral diseases and vector population, thereby causing significant economic losses. Now, in this study, extensive surveys were conducted following field experiments in the state of Chhattisgarh, India. And I explained three archaeological zones. These were the Northern Hill Zone, the Central Plain Zone, and Southern Bastar Plateau Zone, for understanding the distribution pattern of viruses in vector populations in prominent pulses and vegetable crops. Now, field experiments indicated that more than 90% incidence of yellow mosaic disease was recorded in mung bean and erd bean, followed by YMD of ridge gourd and bitter gourd, leaf curl disease of tomato, and chili and yellow vein mosaic disease of bhindi. However, only 5% of yellow mosaic disease was recorded in cowpeas. Now, the molecular and sequence analysis of viral genomic components revealed that the leaf curl disease of tomato in the central plain zone of Chhattisgarh was caused by a consortium of Begumoviruses, including the tomato leaf curl New Delhi virus, the tomato leaf curl Karnataka virus, and the tomato leaf curl Kerala virus. The yellow mosaic disease of mung bean, cowpeas, and soybeans, it was caused by mung bean yellow mosaic India virus, while yellow mosaic disease of ridge gourd was caused by the tomato leaf curl New Delhi virus. And so PCR analysis was done. The use of species to specific primers were used, and the samples representing the three different regions revealed that the yellow mosaic disease of mung bean, erd bean, and cowpea was positive only for mung bean yellow mosaic India virus and was unevenly distributed. So basically, all of this just goes to show that there are several Begomoviruses which not only infect one plant but go on to infect other plants, which in turn has effects on crops. And obviously, it's kind of obvious when we see that, when viruses start to affect crops, what kind of impacts that could have, whether that have an impact on the livelihood of farmers, or whether that have an impact on the economy, etc. Next slide. So now we move on to the management of Begomoviruses. Next slide. So there are several strategies for management. While I've written a lot in my slide, I'll just be going over some of the ones I find most interesting. I thought there are cultural practices we can do, but I thought vector management was quite interesting. Insecticides they're commonly used to manage bee besides the harmful effects on human health and on environment, with the frequent use that led to resistance in the insect and also kills the beneficial insects that are natural enemies of whiteflies. Now, the systematic use of pyrethroids, organophosphates, and nicotinoids has shown the reduction in susceptibility or resistance in populations of V. tabacei in Benin, Togo, and Burkina Faso in cotton. And successes have been recorded with the identification of effective parasitoids of B. tabacei across Africa, and this could work. This could work as a management strategies for Begomoviruses. We have our virus reservoirs as another strategy, which is basically how which is basically, we see that Begumoviruses, they infect weeds and wild plants in the surroundings of sowing horse crops. So to understand the complete epidemiology and establishment of proper management, we need to identify the alternative hosts or the reservoirs, and so that's basically what that strategy is. Next slide. We also have our plant extracts, but I thought breeding for resistance, that was also quite interesting, in the sense that breeding for disease resistance and using a use of resistant planting material, it serves as the best management strategy. However, progress in incorporating durable resistance into cultivars with desirable agronomic and quality attributes, it has significant challenges for breeders, and the use of resistance varieties has remained the most economical and ecologically sustainable control measure. So a promising approach is to breed for disease escape, means to shift cultivation time, which is less favorable for the disease spread. And breeders, they could select for short cropping cycle or for profuse flowering early or late in the season. And that would depend upon the climate of the area in question. Then you also have your virus free planting material. And next slide. Some other strategies we have is integrated pest and disease management, cross protection, which involves protecting a susceptible plant from the infection of a virulent strain. It has been used in the management of some plant viruses already, so it's proven that it works. A Begomovirus mild strain infection of a cassava plant from virulent Begomoviral infection. And plants grown from initially CMD-free parent developed more severe diseases and yielded less than plants derived from mildly diseased parents. And there's also the risk of viruses synergizing to produce a more severe disease. So that's basically what cross protection is. And next slide. And this is the last slide for the management strategies. Basically, we have training and extension education. So that basically talks about how many farmers, they're illiterate. So we need to educate them about the epidemiology and control of Begomo virus diseases, and that would go a long way in helping for the disease management. And the researchers, they constantly receive information through their participation in workshops and other scientific activities. So farmers, they should be trained by being given tips on agro techniques and training, and they should also promote interactions among farmers. They should have orientation programs on epidemiology, and these training camps and extension services in the form of print media, radio, and farmers field schools should also be organized as an agriculture extension. We also have molecular based approaches, which basically we use the process of genetic engineering. We majorly use the process of genetic engineering, and it's been documented as the most effective approach for controlling virus diseases in a wide range of crops grown worldwide. So there's that method. And then we have the Gemini virus database in India, which is being developed at the Modi Institute of Technology and Science in Rajasthan. Now, the objective of this database is to design and provide tools for genetic and in silico analysis of Gemini virus and its genus Begomovirus. And it provides a platform for the current status and research on Gemini virus, especially with reference to Begomovirus infection in different plants across India. So basically, these are all of the management strategies that we can use to deal with Begomoviruses. And the next slide, I think, is references. And that's our presentation. Thank you very much.
Okay, so I'm really speechless. First of all, the Vego virus is very close to my heart. I've been doing work in this area, a lot of work already, and uh, well presented. It was like introduction part and the distribution part, and I love those symptom picks thoroughly. I mean, every symptom so clearly and minutely, I mean, explained. That was great. And Yukta, you explained that life cycle really well. I mean, I loved it. And then the management part was too good because you guys included everything very systematically, right? Right from the, it's like integrated one. You included. You talked about the recombinations. Recombinations are major issues in Begomoviruses. Every now and then we get a new strain of Begomoviruses, right? You talked about losses. You talked about the management, you talked about the molecular approaches, so everything was perfect, right? Uh, uh, I mean, this was definitely no doubt the best presentation of this whole session. Maybe two more are few to come. I don't know about them, but till now this was the best. I just enjoyed it. I just felt that you guys have taken a lot of time to make this PPT, so it was well done, and the references part. So it was a complete package which I expected from you, and you delivered it. All the best, and this just I would say really very good. I enjoyed it, right? And okay, the now let's get to the question and answers from the students. Yes, students, you have any questions? Other groups? Yes, Yuvraj.
Ma'am, I saw some of the slides, and there I saw that there is a term mentioned that AC123, and as I belong to group 2, I just had a question from Yukta. There, can you please specify what does it mean about the AC123?
Yes, Yukta, tell them now. What is this? These are the okay. Let Yukta tell.
So ACE1 to 3 are basically the viral proteins that are required for the replication and transcription part. Like ACE1 helps in the replication. It's a replication initiation protein. It has multiple functions, but here it works as a replication initiation protein, and the ACE2 works as a transcription factor protein which is formed. And the ACE3, I read somewhere that ACE3 is also used for replication, and it has another function that is in host gene silencing, I guess, for ACE3. So these are basically the viral proteins that are
Yeah,
required in
yeah,
the.
yeah. Viral protein which are encoded by the viruses. These are the different proteins basically which are found in the genome of the viruses, and they all have a different functions, right? Okay, anything else? No. Right. Okay, one thing more, students, please upload your PPTs in the file section so that before I give you marks, I just go through them once again, and so that it is available to all the students also of all the groups. So please upload your PPTs in the file section. Yes, Shivani Mishra, yes, go on.
Yes, ma'am. So I wanted to ask, like, can we present our presentation tomorrow in class? Like, we have our presentation on 24, but it's like the last week, so we are left with three
Okay,
internals and some presentation and all.
tomorrow, not tomorrow. Let's do it on Thursday. Thursday we have just one presentation, right?
Ma'am, the problem is on Thursday, Rupal is having her PPT on human value course.
Okay, human value course Thursday. Okay, then your group can present tomorrow. We have a class tomorrow, right? No problem. Which
Yes,
group is this?
ma'am, it's group 5.
Group 5. Okay, no problem. You can present, right? So, since see, Rupal and all other people, since now the standard is raised, right? You can see today's PPT. So, I would like to see your PPTs either equal or more than them, right? So, prepare well. Fine. Okay, students. So I'll take the attendance now, and yes, Adish Mishra,
Present, ma'am.
Athoiba, Ayushi,
Present, ma'am.
Sam,
Present, ma'am.
Devanshi,
Present, ma'am.
Ishita,
Present, ma'am.
Manaswani,
Present, ma'am.
Amrita.
Present, ma'am.
Then we have yes, Amrita के बाद कौन है? Nandini,
Present, ma'am.
Nehal, Rupal,
Present, ma'am.
Aliba,
Present, ma'am.
Anjana, Shakti,
Present, ma'am.
Present, ma'am.
Shivani,
Present, ma'am.
Tanvi,
Present, ma'am.
Sujit,
Present, ma'am.
Vivek,
Present, ma'am.
Yukta,
Present, ma'am.
Yuvraj.
Present, ma'am.
So including the research article data in the presentations make it more interesting, and definitely you get more points because you are reading papers and you are putting someone's research and you are explaining that, because the part that Vivek explained, right, about recombinations and about the problems of these Begomoviruses. So that was really good, guys. We all learn from each other, right? So always referring to the research articles when you present things, it is always a good idea, right? And giving the references. Okay, guys, thank you so much. And it was a really good day for me, at least. Thank you so much.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.